Otherwise, it would still be hard to introduce Caleb. So he's really been a person who's just been outstanding to work with for the last few years. He came in as a mathematician with a pure math background. I once made the mistake of mentioning that he had an applied math undergraduate. He was really upset. And he successfully transitioned into being an exceptional scientist all around. His experimental work is really on par with the finest experimental work that's been done. But let's see, so Caleb, there's actually many Calebs. I think that for those of you who stay for the reception, I think the lab has explicitly laid out the different types of Calebs that are. I want to just highlight a few. So I first met Caleb, the rotation student, which was a transient phenomenon. But in the summer of 2016, he was doing a 9-month rotation with Debbie Marx's lab. And that may seem like a lot, but he ended up getting a really remarkable paper out of that. Which was looking at the 3-dimensional structure of RNA polymers. And at the time, I actually wasn't really looking for any more students. We were a small lab. I thought we were happy. And then Sam, I think it was Sam in the lab, told me that there was this G1 student who I should really talk to, who had ideas. And Caleb came in, it was in the summer, and he was telling us that the way that we were thinking about looking at continuum of states in gene expression was all wrong, and he could prove it to us, but he was going on holiday. And when he came back, he'd show us why we were wrong. And he basically went and he posed just a really nice, he made a simulation and showed that we knew what was going in, and when we plugged it into the visualization methods we were using, we were just getting garbage out the other end. And that really, really clearly showed that things were, and he did this on holiday. And then essentially at that point, it sort of felt like I should really try to engage Caleb. And Debbie and I think, negotiated behind Caleb's back. We'll do 50-50 and so. And eventually, I won. So there's that's sort of the Caleb the rotation student. And then there was then I met Caleb the mathematician, who goes around spouting things like, there's no such thing as pseudotime, and it's not a well-defined concept, and so. But then he goes and thinks about how to pose, to take a problem which is messy, turn it into a clear conceptual problem, come up with elegant proofs, and you'll see, or invoke proofs, and you'll see some of that in his own work. There's Caleb the experimentalist. So the quality of the experiments is very high. Sometimes he gets confused about the questions he's asking. After about a year in the lab, he gave a lab meeting, and he had actually spent quite a bit of time analyzing not his own data, but how many cells each person in the lab had analyzed. And he was very pleased to see that he had actually spent more money than anybody else. Great. Okay, so that's really, those are aspects, and I'm joking, and you'll see the science really speaks for itself. I go now to international meetings, and it's a real pleasure to see people using methods that have come out of Caleb's work now. It just shows the impact of his work has spread very quickly. He's also, I think, many people will know him for other reasons. So he's been a real champion of diversity. It seems a little strange coming from a white guy from Newton. But over his time in his PhD, he's actually taken fairly significant lengths to develop data-driven curriculum on diversity and inclusion, bring in a lot of concepts, and really make it actionable for the PhD executive committee to think about how we're going to incorporate that material, develop a plan. I would also say that there's a little bit of Caleb, the CEO here, because every time that he wanted us to make a decision, he would come up with a document, he would pass it around in advance, there would be clear timelines, decision points. Essentially, there was really no way you could ever say no. You just had to decide which option you liked, and it's a bit scary sometimes. All right, well, I think I'll stop there. There's, without visual aids, I can't show you this. Caleb, the artist, and so maybe he'll show you some of that. Yeah, I should really stop there. I think that generally, a key aspect of a PhD is that you get very knowledgeable with a field. You understand the problems in the field. You understand how to offer approaches to solve those problems, and that you know how to then execute on those problems. And I think that for classical experimental work, it's really clear there's hypotheses you follow up. Often in a new field with new types of data, the problems are harder to define, but I still think that Caleb's PhD will be a really nice example how to pose a problem and then go and answer it. So I'll now give the stage to you. Thanks very much.
Okay, well, thanks, Alon. That's really nice. And I'm excited to say how awesome you are. And okay, so basically, my PhD thesis talk is called A Story of Limits. And so it starts with, as Alon mentioned, my pure math pedigree. So here I am at the college graduation ceremony, and just being in love with math, and I'm so excited, and to just like save the world with math. I think I maybe even had an outsized view of what math could accomplish, which we'll see fleshed out a little bit later. So I entered Harvard really excited to do this, and within the first year I got into a conversation with Alon where he framed to me a problem which seemed like the perfect opportunity to fulfill my dream of like solving the world with math. So I'm going to describe to you the problem that Alon told me about, and then we'll see how well my fantasy played out. So the problem was to understand how stem cells make decisions during hematopoiesis. Hematopoiesis is the process by which blood cells are produced every day in your body throughout adulthood. And it occurs in the bone marrow and is ultimately stems from a population of hematopoietic stem cells which divide and differentiate into the different blood cell types. The way that this process is classically thought to occur is through a hierarchical sequence of decisions. So this figure shows the stem cell at the top, and then it makes a decision to go left or right, and in that moment it chooses one set of fates, it becomes restricted to one set of fates or to another set of fates, and then through a sequence of such steps it ends up in a single final fate. And the way that this model has been produced, at least within this past several decades, is through just a workhorse method, which is FACS and then culture or transplantation. So FACS is where you take cells, stain them by the expression of different genes, and then on the basis of what combinations of genes they're expressing, sort them into different categories. And so those categories might, for example, be the different internal nodes on this tree. Then, once you have the different categories of cells, you can culture them or differentiate them and then see what they have the potential to turn into. So here's an example of the result of one such experiment. Let's say you sorted red cells, green cells, and blue cells. The red cells could only make A, the green cells could make A, B, and C, and the blue cells could only make B and C. Based on this data, you might create a tree that describes hematopoiesis like this. So basically the green cells are at the apex of the hierarchy, then there's a bifurcation where you either get red, which is restricted to A, or blue, which is restricted to B and C. So that's the basic principle by which this tree was constructed. And this tree is in textbooks for the last like 20 years, and it's been incredibly useful for medicine, but yet it's likely not the end of the story. And part of the way that we can appreciate the fact that the tree can be improved is the fact that every year, like every month almost, it seems like there are papers being published with different edits to the tree, and like all of these are pictures from Google Images when you type hematopoiesis, and all of them are showing a topologically different tree. So it leads to the question, like, what the hell is going on? Why is it that people applying the same set of basic experimental tools keep getting slightly different answers from each other? Recently, a set of new experimental techniques are giving some clue about what the issues might be that underlie the different answers that people are getting. So this was a cool experiment published in 2015 where researchers lent DNA barcodes into, in this case it was common myeloid progenitor cells, and then they transplanted the cells into mice and looked at what they became. So basically the idea is that once you take the cells out of the mice post-transplantation, all the cells that share a barcode came from the same progenitor. And what they expected to find is shown on the left, which is that the cells would be purple, as it were. They would be multipotent for both the blue and the red fate, and that you would see combinations of barcodes that spanned both the blue and the red. And yeah, but what in fact they saw is that all of the barcodes were restricted only to blue or only to red. So that means that the situation is probably more like what's on the right, where this common myeloid progenitor state isn't actually bipotent. It's not cells that can actually make multiple fates. It's just a mixture of cells that are already committed to one fate or the other fate. And part of the reason that that might be the case was revealed through single-cell RNA sequencing experiments, where people took these states and when they viewed all the genes and not just the several genes that were used to define the state, they saw that there was actually heterogeneity and that, say, different common myeloid progenitors come in slightly different flavors. So this gives a kind of a lead on what could be going on. These things aren't really the same, aren't unitary. They have differences in their function and also differences in their gene expression state. And in light of this research, a new model has started to be discussed for how you might think about the process. So rather than a discrete sequence of decisions, like what in the classical tree, maybe it's more of a continuum process where cells, they just sort of evolve over time in their gene expression state and they gradually change their fate probability. Okay, so that's the model. And you can sort of appreciate why people might have previously been getting different answers when you think about how we were, if this is in fact the reality, but we were seeing it as a discrete process, this is how we might get different answers. So here I'm showing one way of carving up the continuum into different discrete states. And under this carve up, you might get this as the tree. But on the other hand, if you carved up this continuum in a slightly different way, you might get a different picture of the tree. So this shows that, that to the extent that we're carving up a continuum, that's going to create a limitation on the sort of the real accuracy of our understanding. So fortunately, we do have a way now to look at the continuum, and this is partly thanks to Alon's brilliant postdoc work. And just because this has been discussed ad nauseum in this department, but it's so cool, so I'm just going to play it again. So Alon made a microfluidic device which allows you to look at the gene expression of cells one cell at a time. The way it works is that cells get combined in one channel and enzymes in another channel, and then in the third channel are hydrogels coated or containing barcoded primers, and they all combine in this droplet where cDNA is produced primed with the cell-specific barcode. So on the basis of this data, we can start to create a picture of the continuum, which is a slightly alternative way of looking at it than as a discrete set of states. But here the analogy breaks down, because unlike with facts, the cells don't survive this process. Inside the little droplets, the cell is lysed and destroyed, and you can't see what it does next. And so that was the problem that Alon posed to me, is, or that we were discussing, is how can you take a continuum of single cells and then infer how the cells are moving over time through that continuous state space and what their function is going to be, and thus kind of like create the level of understanding we had before for the discrete model, but now in the situation of the continuum. So in this talk, I'm going to tell you about two different approaches that I worked on to address this problem. The first is a computational approach, and by the title, you can already see foreshadowing that this will not be the final story. And then, therefore, I tried an experimental approach, which was also not the final story, but the story never ends. So, yeah, that's the plan for today. Okay, so first I'll tell you about the computational approach. So the idea was to apply something called the population balance law. So this cube represents a small area of gene expression space. And this is a little bit confusing. It's not a physical space. It's like a conceptual space where the different axes, the different like dimensions of the space represent genes or combinations of genes. And what the population balance law says is that dC/dt, so C here would represent like the concentration of cells as a function of their state. So dC/dt, the rate of change in the number of cells in any given area, depends on the rate that cells enter minus the rate that they leave. So if they enter faster than they leave, then there'll be an accumulation of cells in this particular state. And by making basically an assumption of continuity, this principle can be rendered as this equation, where here, again, this is dc dt, the rate of change. This is a term representing the difference between how fast cells enter and how fast they leave. And then this third term accounts for cell division and cell death, because cells dividing in a particular state is another way that they can enter into that state. So this differential equation, and in this differential equation, this parameter v, that's what we're after, because that says the velocity of cells in gene expression space. It says how they're moving over time. And c is actually what we measure, because when we do single cell RNA sequencing, we're sampling cells from the state space, and that's basically a sample from the distribution C. So bottom line is we have C and we need to solve for V. But now herein lies the mathematical challenge, because, there's a couple other assumptions we have to make this equation well defined, but I just will not discuss that. So the challenge is that this is a wicked differential equation. First of all, it's happening in 50 dimensions, and normally normal solvers can't solve an equation in 50 dimensions. Second of all, we don't actually know what C is, because we're just sampling it, and it's a really sparse sample. So the challenge that we had is there's a really sparse sample, and we're solving a really high dimensional differential equation, so the normal methods wouldn't work. So this was the climax of the beginning. This was oh my god, pure math, it works. Because we eventually found this really cool theorem that had been published by Ting, Huang, and Jordan in 2008, which showed that doing a certain operation on a graph actually solves the differential equation. For people who care, it's that you take the pseudo-inverse of the Laplacian of the k-nearest neighbor graph of these cells. Won't dwell on that. But the point is that this was a really cool, and it seemed elegant solution to solve the equation. And basically the highlight is that the solution you got from this would be asymptotically exact in the limit of sampling infinitely many cells. So you might not get, for any given sample of cell, you might not get the real answer, but as you got more and more cells in your experiment, then you would start to converge to the true solution to this equation. So so then I had a really awesome opportunity to apply this method to a data set that was collected by Sam and Alon, and then our collaborators, Marav and Betsabe at UMass Medical School. And so here is the data set. So this is the octopus figure, and essentially the way to read it is that the head of the octopus is the stem cells, and then each of the tentacles is a low-dimensional visualization of the path that cells take as they differentiate into the different cell types of blood. So for example, one way to interpret this is that this M and GN, monocytes and granulocytes, they have a common progenitor, they're right next to each other in the tree, and you can see that as cells go on a path to M or GN, they kind of hit a fork in the road. And so this low-dimensional visualization is kind of gives us a gestalt picture of how the process is happening. But the gestalt isn't necessarily the real thing. This is just an illustration. So zooming in on this area where all the action happens, what we really wanted to do is get a mathematical description of how cells are moving across this space. And to that end, we applied the population balance law. So here's an example of what it looks like when you apply the algorithm that we did to implement this analysis called PBA. So the idea is that you start with the data, and then you get a map of inferred fate probabilities for each cell. So in this figure, the colors represent the expression of marker genes. So for example, this E is for erythrocytes. That's red blood cells whose whole job basically is to express hemoglobin. And so red is the expression of hemoglobin. But these cells up here that are on the path to becoming an erythrocyte, they're not red because they don't express hemoglobin yet. They're immature progenitors of that lineage. In this figure, now this entire branch is colored red, because in this figure, red represents the inferred probability of becoming an erythrocyte, not the actual reality of being an erythrocyte. So this was the point of why we did this whole analysis, was because we're interested in finding the probability that cells take, because this is a way of answering the question of how do cells move on the continuum. So, all right, so the original motivation for doing this is that we want to reinterpret facts, or we think that instead of being a discrete process, hematopoiesis is actually a continuous process, and that what we used to think of as discrete states are actually regions of this continuum. So the litmus test for knowing whether a method works is that when we sort of take cells and average them back together according to the regions that they were previously defined, that define fact states, we should recapitulate the answers that people got for facts. So that's what's being shown in this plot. So each column here is a different fact state. On the bottom, I'm showing a visualization of the data set. The red cells are cells that we inferred as belonging to each FACSgate by the similarity of their gene expression to what had been published for that FACSgate. And then on the bottom in blue is the actual empirical fate probability of that FACSgate as it was published in the original experiment. And then in red,
The top in red.
so then this is wrong.
No.
Oh, prediction, got it. That's red. And then blue is, what we predicted. So bottom line is that hopefully they look kind of the same. And here is a quantification of the similarity. So bada bang, we did it. We used math to solve biology, and this was amazing, everything I dreamed of in college. Okay, and at this point, I'm going to do something a little bit unorthodox, which is that at this time, I don't know why, because I was just so excited, I just made a movie about this project, and then I never showed it to anyone. And it's on my YouTube page with like a cryptic title of like digits, and as you'll see, the title is still called title page. So I just figured if I don't show this now, it's just gonna be gone forever. So therefore, I'm just gonna show you guys this movie. Every day, 1 trillion of your blood cells die, and your body creates 1 trillion new ones. These cells come in different types. For example, red blood cells transport oxygen, macrophages eat bacteria, and there are many more. All of these cells are produced in the bone marrow by a single precursor called a hematopoietic stem cell. But how does a single stem cell give rise to these very different offspring? The transition from a stem cell to each blood cell type spans multiple cell divisions across several days and depends on different genes being turned on or off. One way to understand this process is to measure the expression of these genes in cells that are transitioning from a stem cell to each blood cell type. Our companion paper measured the expression of thousands of genes in transitioning cells from mouse bone marrow. Here is a map of the transitioning cells as they mature from a stem cell into each blood cell type. The position of each cell in this plot reflects which genes it has turned on. The map tells us a lot. For example, red blood cells, macrophages, and granulocytes are very abundant, whereas the other cell types are more rare. But important information, sorry, just a funny aside, when I first showed this to my dad, I was like ready to be very affirmed for the the work of art, but he just watched the entire video, and then at the end he was like, "You don't say more rare, you just say rarer." Okay. Anyway, there's more. So every day, 1 trillion genes in each blood cell type. Genes, cells, macrophages, and granulocytes are very abundant, whereas the other cell types are more rare. But important information cannot be inferred from this picture alone. For example, this cell is at a fork in the road. What's the probability that it will go left or right? In general, can we predict what all these cells are going to do next? To do that, we would have to see how the cells are moving across this map, which would mean measuring how their gene expression changes over time. Unfortunately, that's impossible, because the device we use to measure gene expression destroys the cells that enter it. To overcome this problem, we developed population balance analysis, or PBA.
PBA begins with a single-time snapshot of gene expression in transitioning cells and uses mathematical modeling to infer how the cells are dynamically changing over time. PBA is explained later.
Okay, or whatever. Then I say PBA is explained at the end of this talk, but that never happened. But, so, okay, that's that. Okay, so as you can tell, I was in a really great mood about this project, and so then Alana and I submitted a paper with a very triumphant title, Asymptotically Exact Prediction of Biological Behavior in in high-dimensional snapshots. And we were really excited. We were like, this is the fake prediction method to end all fake prediction methods. But then the other shoe dropped when we got the reviews. So the reviews, basically the paper was rejected, and the telling review, which after reading this, I was like, wow, that's so true. How do we not realize that? So this reviewer 3 said, thus, I think that claiming exact prediction is highly misleading. What is exact is not the prediction of biological behavior, but only the approximation of one model by another. So just to flesh this out, we had written down an equation. That equation was meant to describe biology. Then we had created an algorithm. The algorithm exactly solved the equation. But there's a whole other step, which is whether the equation is an exact description of reality, which it most certainly was not. And okay, so bad reviews are really upsetting, but this one was especially upsetting because it dashed my undergraduate dream of saving the world with math. Like, so the fear of the mathematician is that we're up in a kind of solipsistic universe, like posing questions that only matter to ourselves. And what this reviewer was basically saying is, you're in a solipsistic universe. Only answering a question that you posed, but what relevance does this actually have to biology? So, Anyway, to preserve our dignity, we decided that if we couldn't solve this